Okay.
So welcome to our webinar today on EuroFlow screening tubes hosted by ALPCO in partnership with Cytometry. My name is Bethany Bell, and I will be moderating the session today. If at any time during or after the webinar you have questions, comments, or concerns, my email address is listed at the bottom of the screen. So just a couple of brief housekeeping items before we start. At any time during the webinar, you're welcome to submit questions in the chat box, which is located in the toolbar on the right. We'll do our best to try to get to everyone's questions at the end of the session. And if for some reason we're unable to get to all of those questions, we will follow up with you with the answer to the questions after the session has concluded. The second housekeeping item, the PowerPoint slides and a recorded copy of this webinar will be available sometime in the next couple of days. When we have that available to you, we'll send you a notice so that you can choose to download them. So here's an agenda of what we plan to cover today during this webinar. First, I'd like to start by briefly introducing you to ALPCO, followed by an introduction to Cytometry, a little bit of a description about what the EuroFlow Consortium is and the goals of the work that they're currently doing. I will pass the controls over to Roberto Juanes, who will then go over the EuroFlow screening tubes in more detail with you. He'll have a brief overview of a very powerful multi-parametric analysis software package called Infinicyt, which was designed specifically to analyze the complex data that is generated using the EuroFlow screening tubes. And then we will conclude with a question and answer session. ALPCO is a developer, manufacturer, and supplier of life and diagnostic tools to research and clinical laboratories. Our products are concentrated in 9 main therapeutic areas and fall under 4 major project types. We offer more than 1,000 human and mouse flow cytometry reagents, including the EuroFlow screening tubes, as well as a powerful data analysis software package called Infinicyt, which again will be briefly discussed during this webinar. ALPCO partners with more than 30 global companies to deliver the scientific solutions our customers need. So our speaker for today is Roberto Juanes. He currently serves as a product specialist for Cytometry. This is a biotechnology company that's based in Salamanca, Spain. Cytometry is dedicated to the design and development of new reagents, software, and techniques that provide innovative solutions in the flow cytometry field. Mr. Juanes was trained in the flow cytometry service at the University of Salamanca, headed by Dr. Alberto Orfao, in one of the laboratories from the EuroFlow Consortium. Roberto, the floor is yours. You will have 40 minutes to go through your slides so that we have plenty of time for anybody who may have questions at the end.
Okay, so thank you very much for the presentation. Bethany, as Bethany says, we'll talk about what is EuroFlow and what is the EuroFlow product line we are offering in the US. And finally, to finish, I will be covering the Infinicyt software. So, well, first of all, I just want to introduce you in what is Cytometry, what is the company I work for. Cytometry was founded in 1996, a lot of years ago, and is a company that is specialized in the clinical diagnosis in oncohematology by flow cytometry. Currently, we have more than 30 highly qualified employees. Most of them, it's PhD or pre-PhD, and we offer global and innovative solutions. That means that we are not offering just reagents, that's antibody combinations, but we are offering the whole package orientated to the diagnosis of leukemias and lymphomas. So we have both reagents and software. We also, because our relationship with the EuroFlow Consortium, as you are going to see now, we consider us as the EuroFlow supporting company. We are really specialized in the whole EuroFlow approach, in the reagents, in the software, all the panels, so we are the EuroFlow supporting company. We are based in Salamanca, in Spain, but we have also office in Rotterdam, and we have a team that covers the whole the whole world, traveling, visiting customers, giving physical support. In that case, I'm the one responsible of the US customers, so any time you need to contact some of our technicians, I'm the person. So what's the EuroFlow Consortium? If you have never heard about it, this is a really good brief explanation about what EuroFlow is. EuroFlow is an independent scientific consortium of European enterprises and research institutions. Well, it started in April 2006 within one of the European fund programs. So the European Commission gives some money to 8 independent research institutions based on different points of parts of Europe, so Spain, Portugal, France, the Netherlands, UK, Germany, Poland, Czech Republic. So European Commission gave some money to these laboratories with the idea of develop and standardize a fast, accurate and highly sensitive flow cytometry test for the diagnosis of leukemia and lymphoma. Also, this European program, this European Commission, needs to be in that project 2 different European enterprises. So one of them was Cytometry. Cytometry has the role within the EuroFlow of develop some software tools for the analysis of the data generated by the EuroFlow approach, as we will see later, and also Cytometry has the rights of develop some of the antibodies that will take part on the EuroFlow panels, as well as the screening tubes, the screening combinations that we are going to see widely today. As I told you, the aim of the whole program was to find a way of make the diagnosis, prognostic, and subclassification and MRD follow-up of hematological malignancies. The general aims, the big general aim of the EuroFlow is one single word: the standardization. The standardization of different things in order to diagnose leukemias and lymphomas. Which things wanted to be standardized by EuroFlow? Well, first of all, sample preparation procedures, setting and calibration of the cytometers, antibody panels, and data analysis. So all these things were standardized by the EuroFlow with the aim of diagnose the leukemia and lymphoma. So in order to do this standardization, they need to evaluate new antibodies, to define multicolor antibody panels, to define all the standard operational protocols, stain of the samples, calibrate the cytometer, all orientated for A-color flow cytometry, and of course develop new software tools and new analysis strategy for multicolor flow cytometry, which is the Infinicyt software. Everything, all the results were published in 2012 in this number of leukemia you can see below, leukemia 26. Also, all papers are free and are available through Cytometry's website, in the EuroFlow website, so you can access to in leukemia website, so you can access all this knowledge completely free, and you can see all the work over 6 years that they did in order to make this standardization. The achievements that EuroFlow has nowadays is the same I told you. After 6 years of evaluating new panels, new antibodies, doing a lot of cross studies between laboratories in order to define the SOPs, they finally, a part of the paper, release, and they are also available in the website, different protocols, let's say, for get this standardization for everyone. First of all is the instrument calibration and compensation standard operation protocols. It's necessary if you want to follow with the EuroFlow to calibrate and compensate your cytometry in a specific way. This is nothing strange. They just wanted to write what everyone was doing in the lab, but just with some tips and some standard procedures, just to have the same fluorescent intensity and the same results independently of the lab that is doing the testing. So thanks to these protocols, if you are following those protocols in the US and applying the same antibody panel, you are going to have same results, completely comparable in terms of fluorescent intensity, in terms of pictures, with another lab that it's in Europe, in Africa, in Australia. So this is because it's so important, the standardization of these protocols. Also, they release the sample preparation standard operation protocols just to make sure that everyone stains the samples with the same time of incubation, same time of lysis, so everything is standardized. And the most important, the antibody panels. A lot of antibody panels for different diseases, acute leukemias, chronic lymphomas, B, T, NK, myeloid, so different panels for different diseases. What is the role of Cytometry in this EuroFlow approach? Well, we have available, a part of the Infinicyt software, the EuroFlow screening tubes. So we are going to see now how is the approach of the EuroFlow, but Cytometry is present in 4 EuroFlow screening tubes, and also we distribute the 8-peak spherotech rainbow that are useful for the calibration, the same calibration I told before of the cytometers. And the most important of all, a part of EuroFlow screening tubes and the spherotech rainbow beads, we also provide with a specialized technical support team. All people working in Cytometry has been trained in one of the EuroFlow labs, the one that is in Salamanca. So we are really used to work with EuroFlow protocols, with labs doing the EuroFlow approach, so we are really trained in order to give support in all aspects, from the calibration of the cytometer to the analysis of the data of the whole EuroFlow approach. EuroFlow product line that we are offering is the same as I just told, is the Infinicyt software one side, of course the specialized technical support, and within the reagents we are offering the 4 screening tubes, LST, lymphoid screening tube, ALOT, acute leukemia orientation tube, PCST, which will come in soon, next weeks, that is plasma cell screening tube, and SST, a small sample tube orientated for small samples. We are going to see these 4 tubes now in deep. We are also offering validated antibodies because the EuroFlow panels are composed by lots of antibodies, so we are offering some of them, and validated combinations. When we are saying that we have validated antibodies and validated combinations, this is because those antibodies are recommended in the EuroFlow publication and also are validating along all the EuroFlow labs around Europe to make sure that they have the quality assured and requested by the EuroFlow. In the reagents, as I told you, we have these validated antibodies combination, and also we have developed new fluorochromes, OC515 and APC C750. One of them, the first is equivalent to Pacific Currents and the second equivalent to APC87, but we want to be sure that we are developing new fluorochromes with more quality than the ones that the EuroFlow used in the past, with more stain index, best separation between positives and negatives, less background. Combining all of these, finally we get with the EuroFlow screening tubes. The EuroFlow screening tubes that are the first step in all EuroFlow approach, is the first tube that we are going to apply if we want to apply the EuroFlow approach. Has 4 different characteristics that are very important and makes the tubes very, very interesting. That first of all, they are lyophilized, so even if it's an 8-color combination, 12-color combination, all antibodies are in the same vial, lyophilized all together, with the aim of have more stability and reproducibility. Tandem dimes, when they are lyophilized, are more stable than when they are in liquid format or separated, and because they are lyophilized and there is no degradation of any fluorochrome, their reproducibility is better. Also, having all antibodies at once, it makes the combination accurate and efficient. All of them, all the screening tubes, are optimized for using with the EuroFlow protocols for an easy integration in the workflow, and in Europe are CE-IVD marked, but in the United States by now are research use only. Doesn't matter, because we are going to see that the screening tubes are, as the name says, screening tubes are for doing the screening, are not giving us a final diagnosis, so you can use it without no problem, even being RUO. You can see now in this slide, this is the EuroFlow flow chart, the classical workflow designed by the EuroFlow people. That this is placed also in the paper, and this is the center of the heart of the EuroFlow. Well, depending on the suspicious that we have, if your sample came to your lab with a suspicious of acute leukemia or a lymphocytosis, or you have a suspicious of some problem with the plasma cells, or you are receiving a small sample too, like CSF or human vitreous or something like that, you are going to apply one of the 4 screening tubes. One of them, a lot, it's for the acute leukemia orientation tube. LST is lymphoid screening tube for mature populations for chronic lymphoproliferative disorders. Then we have the PCST for plasma cells, to screen the plasma cells to see where is the problem, and also the small sample tube that, as I said, is for small samples like CSF. You are going to apply these tubes that are, as I told you, RUO, are not for diagnosis, are just screening tubes, and depending on the result of these screening tubes, you can go one step further and apply one of the different panels that are displayed on the bottom of the slide. So if your screening tube, for example, the yellow T, it's saying that your acute leukemia is B, you are going to apply the B panel for acute leukemias, BCP, ALL. If the screening tube for the acute leukemias is saying that the pathological population, the blood cell, has a T lineage, you are going to apply the T-ALL. The same with the LST, the same with the PCD. So you see, EuroFlow screening tubes are the first step that is going to help us to define which is our pathology, and then we are going to make the final diagnosis thanks to the whole panels. So this is like a 2 steps diagnosis system. First of all, we are going to apply this screening to know what is happening and to discard normal samples, and then we are going to apply second step just to make the final diagnosis. We are going to see these 4 screening tubes in detail, combination of antibodies, formats, everything, just to see the advantages. First of all, the LST, lymphoid screening tube. This is designed for the study of B, T, and NK mature populations. Even if it's 8 colors, we are putting 12 antibodies in the same combination. Why? Well, because we are combining in the same color, in the same fluorochrome, different antibodies that are exclusive, so are for different lineages, different populations. You can see, for example, in the Pacific blue, we are combining CD20, that is for B cells, and CD4, that it is for T cells. So we are not going to have, at any moment, any population with co-expression of 20 and 4. And we have adjusted also the intensity of the fluorochromes for each antibody to make sure that the 2 populations are perfectly separated. Also, the same thing happens in FITC, where we have CD8 and lambda, for P, when we have 56 and kappa, for TCR gamma delta, when we have 19, for P7, we have CD19 and TCR gamma delta. The rest of the combination, you see that we have markers for general lymphocytes, like CD45, we have for T, 3, 4, 8, and 5, we have for B. Also 4 and 8, and we have for B 19, 20, and 38 to discard plasma cells. So those are the suspicious that we have with when we are going to apply the LST with, for example, monoclonal component, or unexplained cytopenia, or unexplained eosinophilia, lymphocytosis, lymph node enlargement, or atypical lymphocytes, or splenomegaly. If we have those aspects and we don't know what it is, we're just going to apply the LST to see if the problem of the patient is B population, T, or NK. Depending on the result of this screening tube, we are going to move to the whole panels composed by several antibodies, BCLPD, TCLPD, or NKCLPD. And then we will make the diagnosis. But thanks to the LST, we can orientate our study to B, T, or NK. Here is what happens when we are combining the analysis with Infinicyt and the LST. The LST in the left side is a bunch of normal samples. There are, in fact, 4 different normal samples that we have put all together and analyzed, and we can see the expression of the normal population. So we are doing multi-dimensional analysis, and we are separating all different populations that are in the sample, so NK, T, 4 and 8, double negatives, kappa, and lambda cells. So this is for 4 normal samples, all populations separated. What happens if we are applying an LST over a new sample that we don't know what it is? we can see how all populations fit in the same position than the normal, but we have some pathological B population that is outside of what we consider in normal. It's near B cells, so doing this analysis we can conclude that my pathological population is B. Then I'm going to apply the B panel just to define if it's a CLL, a Burkitt lymphoma, follicular, whatever. The LST has a high efficiency in order to discriminate if my pathology is BT or NK. It's proof in the paper that I mentioned before. You can see that information. The overall efficiency in discriminating if it's BT and NK is 97.4% of the cases. In 99.4% of the cases, if it's B, we are going to know thanks to the LST, and in 94% of the cases, if it's T or NK, we are going to know also. The left percentage of uncertain results is when there is some diseases that cannot be easily distinguished or by phenotypical, some things like that. The LST can be used in peripheral blood, lymph nodes, bone marrow, also thin needle aspiration, so can be widely used. For small samples, we are going to see that it's better if we apply the SST, that is the one that comes after the LST. The LST, well, at the beginning I said that the EuroFlow antibodies were validated. Well, how is done this validation? To do the validation of our LST combination, we have an internal and an external validation process where we merge together, we put together several normal samples. We made this multidimensional analysis where all populations are separated, and then we plot on top of these normal samples a new combination that we are testing. If our combination is the expression of all the antibodies is similar to what EuroFlow is proposing and what EuroFlow is requesting in terms of fluorescent intensity, in terms of separation, in terms of quality instead of staining this background. If our antibodies are exactly the same with the same quality of EuroFlow requesting, our new combination is going to be plotted on top exactly in the same position of all samples that we have in-house and we have also in the different EuroFlow labs. If we are not completely sure that our combination fulfills the EuroFlow requirements, we never release that combination to the market. I explained this with the LST, but this validation process is done with all antibodies belonging to the EuroFlow panels and all screening tubes. What is the format of the LST? Well, at the beginning I said that all EuroFlow screening tubes were in lyophilized format. So we are offering this LST in a kit of 25 tests, but these 25 tests are splitting in 5 different tubes of 5 different tests, as you see in the picture. Why? Well, to make sure that we are fulfilling all labs' needs. Some labs will not need so many tests as a vial of 25, and some of them will need maybe 5 a day or 5 a week. So depending on the volume of your lab, you are going to reconstitute only one vial or all of them. This is because we split these 25 total tests in 5 different tubes. Because we are introducing also some new fluorochromes, as I told you at the beginning, like the OC515 or the APCC750, and also the tubes include a tandem of PCyanine7. We include in the kit a vial for each of these antibodies to make compensation. So if you need to make a new compensation in your cytometer to adapt all voltages and all compensation to these new fluorochromes that we are introducing, we are including in the kit these vials for the compensation protocol, so you don't need to use the LST indeed. So this is the format what we are offering. So it's 25 kits split, 25 tests split in 5 different vials of 5 different tests, lyophilized, so 12 antibodies, all combinations together in the same vial, and also antibodies to do the compensation in case you need to do a special compensation for using the LST. The advantage of having everything lyophilized, of course, is that you just need to pipette once to add 12 antibodies at the same time. The quantity of the antibodies is adjusted to make sure that all of them are giving the best signal, as I told you, according to the EuroFlow standards. Also, fluorescent intensities are standardized, and because it's lyophilized, all tandems are stable. The stability of the lyophilized kits, when they are still lyophilized, is 1 year, and when they are reconstituted, it's 1 month. Next tube is SST. It's like a variation of the LST. In this case, instead of 12, we have 11 antibodies, and it's designed for the study of small samples, like vitreous biopsies, CSF, vitreous tumor. So every time that you have these kind of samples in your lab, instead of applying the LST or the ELOT, we are going to apply the SST, the small sample tube. This tube has antibodies for lymphocytes like CD45. We have also for T like CD3, 4, and 8. We have also for B cells like 19 and 20, kappa and lambda. We have 56 for NK, and we have CD14 for detecting monocytes. Also CD38 to see if we have some plasma cells, some infiltration. Okay. So thanks to this tube, we are able to detect the most relevant leukocyte populations in low cellularity samples. Higher specificity and good sensitivity better than cytomorphology. This can be also consulted in the paper, all the studies that they did doing a cross study between different techniques. Okay. So as I told you, we can see, thanks to this antibody combination, infiltration of lymphoid populations, BTNK, plasma cells, and monocyte in this kind of small samples. This is a powerful tool for the evaluation of samples with primary lymphoma suspicious, and thanks to the multivariate analysis that we have seen in the LST, the diagrams within Infinicyt, called APS, helps to discriminate normal and pathological populations. You can see here in the picture an example where we are able to distinguish in a pathological cerebrospinal fluid sample stained with the SST, T cells. We can also distinguish the CD4 and CD8, monocytes, and a pathological population. Doing multivariate analysis, that is the diagram in the right side, we can distinguish clearly and without doubt all populations that we have selected in the other diagrams. How is the format of the SST? Well, as the combination is similar to the SST, all markers are surface markers, we can offer exactly the same approach that we did in the LST. So a kit composed of 25 tests divided in 5 vials of 5 tests each, and also including the vials for the compensation of these specific, antibodies. Next tube is the acute leukemia orientation tube, a lot. In this case, we have 8 antibodies, 8 colors. Okay, so there is no more than 1 antibody in one position. And we are going to apply this acute leukemia orientation tube when we have a suspicion of acute leukemia, of course. You can see that we have antibodies for defining mature and immature cells, like 45 and CD34, and then some antibodies for the initial evaluation of B lineage, T lineage, or myeloid lineage. So we have CD3, both cytoplasmic and surface marker. We have MPO for the myeloid lineage, and we have 17A and CD19 for the B lineage. Okay, we are including both CD3, surface and cytoplasmic, because in some acute leukemias, not all molecules of CD3 are outside, are in the membrane, and we are not sure that is expressed in CD3. So this is because we include the cytoplasmic CD3, also with the MPO and 79A. Using the acute leukemia orientation tube, we are going, and depending on the result, we are going to apply one of the 3 different panels for acute leukemias that are the BCP-ALL, T-ALL, or for AML. The efficiency of the ALOT to discriminate if your acute leukemia is T, B, or myeloid is 98.3. The rest of the percentage, the 1.7, is for the ambiguous lineage when we have some phenotypical acute leukemias. But for the BT and myeloid, the efficiency is 98.3. So we are going to be completely sure that applying our allotute, we are going to discriminate the lineage of our leukemia. Here is we can see a diagram with applying the allot. We can perfectly distinguish cases of AML despite of the BCP ALL. Also, it's completely possible to discriminate TLL cases of BCP and also AML of the T lineage. Once again, all this information is present on the paper, so you can see all the results and all how many samples they use, everything. Here we can see an example of how to use the allotute in combination with multivariate analysis within Infinicyt software. We have merged together and put together different cases of AML, of BCP ALL and TLL. The T's are in green, B's are in blue, and AMLs are in orange. Okay, so we have put lots of cases of one of each diseases, and thanks to the multivariate analysis with Infinicyt software, we can clearly discriminate all of them, all B's, all T's, or myeloid. When we have a new sample that we don't know what it is, applying the allot combination and gating our blast cell population with 45 and 34, if we plot this population against all of our reference, the multivariate analysis is going to indicate that our pathological population fits in the same position, so has exactly the same expression of all cases that we have called BCP ALL. So we can be sure that applying the allotute in combination with the multivariate analysis that Infinicyt is offering, we can have the orientation to the B panel. So you just need to use the allot combination, gating on 45-34, and then the multivariate analysis is going to give you which lineage is present in your blast cell population. How is the format of this allotute? Well, it's slightly different of the LST and SST for one single reason. In this case, as you can see in the combination of antibodies, we have some surface markers and some cytoplasmic antibodies. So we cannot put everything together in the same vial because the protocol is slightly different, so we need to split. So what we are doing is put together all membrane staining antibodies, put together all cytoplasmic staining antibodies, and then split it. So we have also 25 tests, and we split in 5 different tests, but we have 5 different tests for membrane and cytoplasmic, 5 different tests for membrane and cytoplasmic, so on. We include also compensation reagents, and we include also the Nordic Movio fix and perm reagent, A and B solution for fixation and permeabilization. So you can do the whole protocol of staining with our kit. You don't need to get the fix and perm from other suppliers because we are including that in the kit, so you don't need anything apart of the allot kit to make the staining of the sample. Last tube I want to talk about is the PCST. Is the plasma cell screening tube. It's not available yet, but it will be in a couple of weeks. In this case, it's also 8 antibodies and is designed to the study of, as the name says, plasma cells. So it's for the identification and enumeration of normal and aberrant plasma cells. It can be used as an initial screening of plasma cell dyscrasias like multiple myeloma or MGUS, and also depending on the result of this tube, if you apply following tubes on the EuroFlow approach, you can do the MRD follow-up. So when you have a monoclonal component, not IgM or bone lesions or bone marrow plasmacytosis, you can apply the plasma cell screen tube. Which is the combination? We will have 145, 138, 38, 56. Also for prognosis purposes, we have beta-2 micro, we have CD19, and also intracytoplasmatic kappa and lambda. As you can see, because we have cytoplasmic kappa and lambda, the format will be similar to the allot tube, but we will see at the end. Thanks to the PCST, and a unique immunophenotypic profile, we can distinguish with this tube between normal and clonal plasma cells. The backbone markers that we have for the distinguish of plasma cells are the best one selected by the EuroFlow, so 38, 138, and 45. And then thanks to the additional markers, we can characterize these plasma cells, 19, 56, the light chain restrictions. So you can see in the picture how we can distinguish with Infinicyt in normal dot plots and then with multivariate analysis the normal and the pathological plasma cells, those populations that are PCST designed for. So you can see that when we are using normal dot plots, it's easy to distinguish normal and aberrant plasma cells, but thanks to the multivariate analysis, right side, so APS, multivariate analysis, we are really sure that we are distinguished perfectly fine normal and aberrant plasma cells. As I told you before, the PCST has a similar format of the allot tube for exactly the same reason. We have some cytoplasmic reagents, in this particular case kappa and lambda. So we need to split these 25 different tests in 5 vials, but one of them for cytoplasmic and one of them for membrane. We also include the Nordic Movio fixed and perm reagents for fixation and permeabilization, and as usual, all compensation reagents needed to do the compensation for these special fluorochromes. Just to summarize what we are seeing today, it's our EuroFlow product line that we are offering. You can see that it's completely related, that from one side EuroFlow reagents, screening tubes, LST, allot, PCST, and SST. Just try to remember that those tubes are with the aim of do the initial screening of your sample. The aim is not the diagnosis, it's just to make a screen and then, depending on the result, apply a particular panel. So depending on the result of the acute leukemia orientation tube, you will have a panel for leukemias, depending on the LST, a panel for chronic lymphoproliferative disorders, and so on. So we are offering the first step of the EuroFlow approach. Advantages: lyophilized format, so more stable, easy to use, not a lot of pipetting, just once pipetting in the case of LST and SST, and 2 pipettings in the allot and PCST. Everything is validated, so we have the validation by the EuroFlow, so our reagents are getting the desired quality, the desired fluorescent intensity, not background, etc. We have seen how the combination with the Infinicyt software is essential for do the analysis of these screening tubes, and how easy it is to have a final result using Infinicyt, and absolutely the specialized technical support. Just to finish, a few words of the Infinicyt software, just to have it more in detail. EuroFlow was designed within the, Infinicyt was designed within the EuroFlow project because they need some special tools that were not available at the moment in the market when they started with the project in 2006. So they decided to create a new software from scratch, including all new tools, all multi-dimensional analysis that they were not present at that moment in the field. They face that they have new technology. At 2006, 8 color flow cytometry started to raise in the market. Also more new fluorochromes, new antibodies, new clones, so they have a lot of new technology, but they face that they have more problems. They have more files, more diagrams, and of course the interpretation of these 8 color combinations were really difficult. So they decided to create a new approach for the analysis, it's multivariate analysis, and put it within a software that it's the Infinicyt one. Advantages of the Infinicyt is that it was developed within the EuroFlow project, so it's directly related with all EuroFlow panels and approaches. Of course, Infinicyt, it's open to any kind of reagent, any kind of panel, any kind of studies, research, clinical, so there's no limit with Infinicyt, but because it was designed in the heart of the EuroFlow consortium, it's working perfectly fine with all this EuroFlow approach. It's a new concept. It's not classical analysis, 2x2 plots, histogram. It's something that's different. It's a multi-parametric analysis. I mean, we have everything that other softwares have, all classic analysis you are used to, but also we include a new step, a new differentiation with the multivariate analysis. Infinicyt is ready for database integration. EuroFlow will release in the near future. We don't know when, and we don't have that information, but we know that will be in the near future some databases of reference where you can compare, if you are following the whole EuroFlow approach, you can compare your cases against all cases gathered by the EuroFlow all these years. Infinicyt is already ready for that now. Even you can create your own databases of reference if you have enough cases. So if you have the power to do that, do not hesitate. We are offering the support to do that, and you can compare then your cases against the database. There are two kinds of database cases. First is Compass tool designed for the chronic lymphoproliferative disorders, BTNK, so all these panels, BCLPD, TCLPD, and NKCLPD, and also maturation tool, where we are studying maturation patterns in bone marrow for the study of MDS. We include, and this is really important within the Infinicyt software, EuroFlow profiles, so all the templates required for the analysis of the 4 screening tubes are included within Infinicyt. So if you are using our screening tubes and Infinicyt at the same time, you have all profiles, all templates with all diagrams, strategy approaches, everything set up. Just need to load the file and start with the analysis. You don't need to configure anything because everything is included within Infinicyt. This is an example of how is the comparison of your data with the databases. So at any moment that you have a new case that you don't know what it is and you are following the whole EuroFlow approach, you can just tell the software, please compare this case against all cases I have in the database, and it will give you an orientation about what is your case, if your case is a disease, a group of reference, or another. This is for the chronic lymphoproliferative disorders, and this is for the patterns of maturation. So for example, when you are studying bone marrow in MDS, you can define normal patterns of maturation from stem cells to mature population in neutrophil lineage, erythroid, monocyte lineage, and then save these reference patterns and compare your results against these patterns. This is just a few approach of what Infinicyt is. But I just want to make clear that it's a software that works perfectly fine if you are following the EuroFlow approach. The screening tubes is ready to connect with all these screening tubes and make your life easier if you go for the whole approach. Just to say thank you to Alco for giving me the opportunity to present you today this product line, the EuroFlow product line that we have, reagents, software, and technical support. And I think that it's time for me to stop and give the word again to Bethany, that is giving you some important information now. Thank you very much.
Roberto, thank you for a great overview of the topic. I hope that everyone found it to be very informative. If anyone would like additional information on any of the products described by Roberto, we would like to encourage you to visit the resource pages available through both Cytognos and ALPCO. The web addresses are listed here. We will be attending the listed trade shows, and we would love to meet you and talk more with you about the screening tubes and how they may improve workflow in your lab. Those meetings, denoted by an asterisk, are open for anyone who would like to schedule private training in their laboratories to see how their workflow could be improved by implementation of these screening tubes. So definitely reach out to us if that's something that you would like to start to set up now. So at this time, we'd like to open up the Q&A session. As mentioned in the beginning, you can enter any questions in the chat area on the right of your screen. So we welcome any questions that you have. So the first question, and this is a workflow question for Roberto, how long does it take to run the EuroFlow protocol from start to finish?
Okay, well, if you want to run from the beginning to the end of one diagnosis panel, not the end of the screening tube, it will take, depending on how many, because the EuroFlow panels, not all of them has the same number of tubes. So after the screening tubes, we have panels of 2 different tubes. We have panel of 7 different tubes, so depending on the number of the tubes. But for an average panel of 5 different tubes, and after all the staining, it will take a couple of hours.
Does the
So first step,
EuroFlow protocol require a specific instrument?
well, EuroFlow was designed at the beginning for, of course, you need an 8-color or plus instrument, but first was designed for BD Canto 2, but also now the own EuroFlow consortium is doing validation studies with culture machines, and we know that in few months all these studies will be released, and all protocols will be also adapted to the Navios machine.
The next question is, can Infinicyt be used to analyze data for antibodies other than the EuroFlow screening tube?
Yeah, absolutely. As I told you, Infinicyt is open to any file that comes from a cytometer. It doesn't matter if it's EuroFlow or not. We have customers doing research, doing EuroFlow, doing clinical diagnosis, not EuroFlow, so it's open to any file that came from a cytometer.
Is there a demo available for the software program?
Absolutely. You can visit infinicyt.com and you can download a demo version available for 1 month.
Okay, does anyone else have any questions? If you do, go ahead and enter them in the chat box on the right-hand side of your toolbar. Okay, it looks like that's everything. I would like to mention again that the slides will be available in the coming days. My contact information is listed here if there are any additional requests. Roberto, thank you again for an excellent overview of the topic, and to everyone for joining us today. At this time, we will conclude the webinar. Everyone have a great day.
Have a great day.